So today I would like for us to cover chapter 13, not the vocabulary specifically, but all of the rest of chapter 13, the concept portions. I think you guys would see on the module, there is a note outline. It's 2 pages. It's an outline. It doesn't have every single bit of detail in it. It leaves a little bit of space for you to add definitions or comments as we go along. As well as it just kind of gives you a guide of the important, hitting the high points of this chapter. The vocabulary we've already spoken about, putting the words on cards, on vocab cards, flashcards. I highly recommend it. I highly recommend putting words that are related to each other together on the same card so that you have fewer cards to flip through and sort through. I will be asking, I believe it's 30 of the vocabulary words as matching. Now, from doing this in the past, I have found out that I cannot just load 30 terms and 30 definitions as I would on a regular in-person paper test. So they'll be blocked out probably in groups of about 10. So there'll be 10 terms and 10 words 3 times. And there will probably be a few distractor words in there, extra terms, but all the words will only be used once. As I mentioned before, but I'm going to mention it again because this will be a longer recording and hopefully more people will take a look at it. I will be using the definitions in context, meaning the definitions from around the words. I will not be using the full sentence definitions from the index or glossary, and I will certainly not be using the definitions from some various online source. I will be using the definition in the context, and if you, in theory, of course, if you look up the definition somewhere else, you should be able to figure out the definition in context when I give it to you. But my reason I'm telling you this is keep it simple. Just learn the definition from the words around it in context, and then you'll be able to answer those questions a lot more quickly than if you had a different definition and had to figure out what part of it was similar to the definition that I was using, right? I'm telling you straight up. There's like 90 some words. I'm going to use the in context definitions, the words around the word in the textbook. So you're going to have to read the book. All right, so enough about the vocabulary words. Let's start taking a look at some of the content, the important content, high points that I need you to pay attention to for this chapter. And those of you that have already checked this out in the module, you'll be well aware that there is this set of PowerPoints in the module. And I'm going to try to hide the ribbon. There we go. And make it as large as possible. So I'm hopeful that you can actually see this. Yes, can somebody just give me a yes, we can see it? I know sometimes I start to talk, especially in BIO1, and they don't seem to be able to see what I'm doing. Yes, okay, awesome, thank you. So the current understanding of where there are sites, and this is limited to humans probably, right, where they contain normal microbiota. And what I want you to note here is that, of course, in the respiratory tract, digestive tract, reproductive area, the ear canal, and your eyelids, your eyes and your eyelids, of course, could harbor microbes. But these are some new areas that we used to say were sterile or did not have any microbiota. And now we know that they have at least some: lungs, bladder, breast milk, amniotic fluid, and even fetal fluid. Sites with which DNA has been detected, both the brain and the bloodstream. So DNA is detected. So be careful there, because that means we know that the DNA is there, but we don't have evidence the whole organism is there. So that could be just DNA from a destroyed organism, but we don't know, right? And again, these both we would have previously thought, especially the brain, we thought was sort of a privileged area where there weren't any bacteria or viruses, not many, if any, entered. And certainly there weren't any that were what we would call normal microbiota. Now remember, normal or resident microbiota, we acquire those when we are babies, as soon as we are born, and we continue to acquire them throughout our lives from the environment, from people that we touch, from the food that we eat, be it from a bottle or from breast milk. And I'm not asking you to know what bacteria are in these sites. I just want you to have some general idea. Normal microbiota, we would say, are microbiota that are common to many people within the human population. We've talked a little bit about the Human Microbiome Project, but I do want to bring it up again because what has been discovered, and this is just like new and fascinating and really exciting for those of us who are microbiologists, is that even places like the skin, which we would normally, like when we get to chapter 18, I'm going to give you a list of organisms that are normally found in the skin. What you're going to see is that list is quite expanded from what it used to be. And what we know from this microbiome project is even an individual person, so like anybody, so I'm just going to use myself as an example, I'm left-handed versus right-handed. So because I'm left-handed, my dominant hand has actually some different organisms on it than my non-dominant hand, and a different amount than my dominant hand, my non-dominant hand. And it's not even like that. You might think, oh, well, that makes sense because you touch a doorknob. If you're left-handed, you touch the doorknob with that hand, or you write with that hand, you pick up a pencil or your car keys with that hand, or your phone with that hand. But even more than that, let's say we're talking about someone's arm. What they've discovered is even from this side of my arm, like the inner side of my arm, like inside my elbow or inside my forearm, compared to the outside, different bacteria, sometimes completely different bacteria. And that's going to vary all over different sites in our body. And what we used to consider normal flora or resident flora that we thought were microbiota, we call them now, we used to call them normal flora, that are with us all the time, many more of them are transient than we thought previously. So these contribute not only to the overall health of our bodies and us as an organism, but also can influence when we get sick, because sometimes it's an imbalance between the organisms that normally inhabit us and an invader or a pathogen organism. So I just wanted to bring that up because I still find that interesting and fascinating. And I'm going to go back and share some more. So, okay, so we talked about the sites containing normal microbiota. This is important. This is steps involved when a microbe causes disease in a host. And there's some vocabulary here and some ideas that we need to talk about. We refer to how the organism gains access as a portal of entry. And I did list on your handout some portals, some common portals of entry, and you're going to see they match pretty much here. Skin, gastrointestinal tract, I put more generally the mucous membranes, and endogenous biota that accidentally get inside of us. And I put on our list parenteral, which is something that I'm going to start a second share here so that I can define that word for you. So parenteral, which is not on the list in your book, that means bacteria or microorganisms that are injected directly into the bloodstream. So under the skin, directly into the bloodstream. And so these would be deposited there by a puncture, like a needle, as well as a deep cut, an animal bite. I've listed there dog, cat, snake as some examples, right? But some way that the organisms get deposited directly under the skin, which again, that is infrequent, but it is a way that organisms can actually get inside the body. So they have to have a portal of entry. And then again, on the handout, you'll notice I listed ID50 and LD50. I believe those may or may not be in bold. They might be in italics. The ID50 stands for infectious dose 50%. ID, and the 50 is usually subscript, but I can't do it here on this keyboard. Infectious dose 50%. And what that means is not 50% of the bacteria or the organism. We're talking about what is the dose necessary to infect 50% of the test population. And usually the test population is not humans, unless we get it from a retrospective study, it's usually done in an animal study, and it's detected about how many organisms do we need to infect 50% of the test population, and then that is extrapolated out to humans based on our size and the, how many bacteria would need to be to infect 50% of people. The LD50, that stands for lethal dose 50%, and that's how much do we have to ingest or get into our mucous membrane to actually kill 50% of the test population. And again, this would not be something that is actually tested in people. This would be something that would be tested in an animal model and then extrapolated to people. And you might think, well, what do we care? Why do I have to know about the ID50, the LD50? And I would remind you, think back to chapter 7 and think back to when we were talking about counting how many bacteria were in a water sample or a sample of milk or beer or apple cider. It's kind of on my mind because of the season that has been pasteurized, but you still need to know how many bacteria are in there, and if the number of bacteria is below the ID50. So for example, there are standards like for pasteurized cider, pasteurized milk, pasteurized fruit juice, that there is an ID50 and it has the organisms that you isolate from your post-pasteurization process. And you have to show how many were in it pre-pasteurization and then post-pasteurization, how many bacteria, and correlate those to the indicators of what is an acceptable number for that being below that infectious dose 50%. The lethal dose 50% is usually not a tested dose. It is usually an inferred dose based on statistics, based on some epidemiology studies. But the lethal dose is usually a bigger dose, larger dose than the infectious dose. That makes sense. And I don't think this is in our textbook anymore, but some examples are like for some bacteria, you may only need 50 to cause an infection, and other bacteria you might need 500,000 to cause an infection. So you can see there's a great range of what that infectious dose 50% is, and it's different for different organisms. And so those are numbers that people who would be involved in testing food and water would need to be aware of. All right, let's go back to the PowerPoints here. There are some mechanisms for adhesion, because you'll notice I listed the next thing in the progress of an infection is not only does a microorganism have to get in, it has to be able to stick and stay. And this chapter is all about the structures, the chemicals that the microorganisms produced to overcome the host defenses. And then the next two chapters are going to be all about the host defenses and how they can overcome the microorganisms while they're trying to infect. So be aware that this is sort of a shared, bacteria are trying to stick, your cells are trying to prevent them from sticking. In this chapter, we're focusing on if adhesion occurs successfully. And I would remind you some of the structures that help bacteria stick or adhere. Would be if the bacteria have fimbriae, if they have a capsule, and of course viruses with their spikes that are specific for receptors on the host cell. Once the organism attaches, the organism, particularly bacteria, can do some direct damage by creating some enzymes that either break down or damage tissues, or they attack some of the host, particularly usually the host non-specific defenses, and help the microbes spread into deeper tissues or get into your bloodstream. And here's a list of examples of some of the exoenzymes that, bacteria in particular may produce. Notice on the handout I wrote not all bacteria can produce these. Most bacteria only produce one. Some super virulent bacteria may produce more than one of these. And I think the list is a little bit longer. Oh, excuse me a second. Hello? Hello? So these enzymes here, notice they all end in ASE. And again, most of the time they're going to tell you specifically what it's an enzyme, right? So enzymes usually give you their name and they tell you what they do, right? So for example, mucinase, that's a very generic enzyme name that's going to break down mucus, right? Collagenase, that's a lot more specific, and that is going to particularly break down collagen. Now, that's really important because if a bacteria is able to break down collagen, then they're going to be able, if they have collagenase, they're going to be able to break down collagen, which is obviously really important connective tissue in your body. And that's going to help the bacteria spread very quickly and certainly into deeper tissues and cause way more serious damage than something like just a mucinase, for example. Let's see, as I look down the list, I would pick out coagulase as an important one. We'll see that one show up again with the bacteria, a lot of the bacteria that infect the skin and some that infect respiratory and cardiovascular tissues, because they're going to cause coagulation or actually break down clots in the blood, right? And then we have kinases, homolysins, they're going to break down red blood cells. Oh, I think I put that picture in, so let's show that picture. Here's one that's, here's a picture that's showing you the different types of homolysis by different bacteria on blood agar. So these two bacteria do what we refer to as beta hemolysis. So they secrete hemolysin, which breaks down the red blood cells completely. These bacteria, they produce nothing, and this one produces kind of a greenish cast because it partially breaks down the red blood cells. Good for the bacteria, bad for the host, right? The bacteria can destroy the red blood cells. They can limit the amount of oxygen that's being delivered to a particular place in the tissues. Many of these bacteria are aerotolerant. So think back to chapter 7. They would prefer to be where there is less than 100% oxygen, right? They would prefer aerotolerant. They grow better when it's anaerobic or closer to anaerobic conditions. So those kinds of bacteria would produce hemolysin. The hyaluronidase, I do like to mention that one specifically. There should be a comma here, or more of a space. But the hyaluronidase, that is going to break down hyaluronic acid, which of course is another important component of connective tissue. And then some bacteria can produce very specific toxins. We title them exotoxins or endotoxins based on their characteristics. Characteristics are listed here in the center of the table. And then the differences between an exotoxin and endotoxin are highlighted out here to the left and to the right. Again, not all bacteria can produce endotoxins. It's some bacteria can produce them. Exotoxins in particular are related to, they are released outside of the bacterial cell. Exo, they are released or secreted from the bacteria, and it's pretty much usually from a gram-positive bacteria. Very few gram-negative bacteria. These exotoxins are super specific though. They actually bind to very particular places inside the host, and they only need to be in small amounts, and they can cause death. The advantages, though, they are what we call unstable or heat labile, meaning that you can, if you heat them up to over 60 degrees, they denature, and then we call them a toxoid, and you can actually use the toxoids as a vaccine, which is in the manner of the DPT vaccine for diphtheria, tetanus, and pertussis. Endotoxins are a little more general. They are not super toxic. They usually produce a fever because they are part of the cell wall. They are the lipid A portion of lipopolysaccharide, which is in that outer membrane of a gram-negative cell wall. So you'll notice that it says all gram-negative bacteria here can produce endotoxin. However, endotoxin, it is part of the cell. It is not released until this bacteria dies. And when this bacteria is broken up and torn apart by host defenses is when the endotoxin is going to be released. So it sometimes happens late in the infection when a person might seem like they're almost better, and then they get an increase in fever and an increase in this release of endotoxin can really cause someone to feel pretty grody. Then I also listed on there, when colonization leads to disease, I also listed on your handout here, and I'm checking just kind of briefly with what's in the text. I don't know why I put that on there. Causing overreactions of host defenses. I think particularly we're talking there about the endotoxin, and then epigenic changes in host cells. We're referring to when a microbe has the ability to regulate part of the DNA in the host cell. And this is usually small RNAs, small regulatory RNAs, and these changes can sometimes permanently affect the host and the DNA in the host. So I'm going to list that. The next thing I wanted to talk about a little bit was so here we've talked about the sort of the beginning of an infection. This is a classic curve that marks what we call the stages in the course of infection disease, or the stages of a disease, or you might hear it called the course of infection in your nursing textbooks later on. And these periods, incubation, prodromal, period of invasion, convalescent, and continuation period, these periods all have very specific definitions, and I wanted to run through those with you real quickly. So the incubation period is the time between, notice time is on the x-axis here. This is the time from your initial exposure to your earliest sort of non-specific sign or symptom. This might be until the first time you feel like, oh, I don't feel so great today, or I'm a little hot, or I have a headache. This prodromal period is usually 24 hours or less, and that's when you experience your first mild signs and symptoms. So this is the time when you might not feel 100%, but you wouldn't really necessarily think that you're sick or have an infection yet. It's usually very difficult to diagnose an infection during the prodromal period. This next phase, period of invasion, we used to call it period of illness. It is also referred to as period of infection, right? This is when you exhibit all the classic signs and symptoms of a particular infection that would help a physician or a nurse help diagnose what exactly infection you have. So, for example, if fever is considered a sign because it's a measurable change. Anything that ends in itis or ache is a symptom, right? Those are subjective feelings. But for example, there are some classic, for like the measles, there's something called Koplik spots, and they show up during the period of invasion or at the height of infection. And they are white spots that appear on the inside of the mouth of a person who has not yet expressed the measles rash. Okay, so they're an early, and they are a sign. They're a clinical sign because they're very distinct. Period of invasion, usually there's a range of time here because, again, some people, if you are particularly healthy, your period of invasion or infection might be smaller. When you're younger, this tends to be a shorter period. As you age and your immune system ages, it tends to be longer. Or if you are battling two infections at the same time, that period might be longer. This period is super important, the next phase. It's called the convalescent period. This is the time when you are returning to pre-diseased state. This period is longer than most of us think. Right? This is the period where we're like, oh, I've been so sick all week, but now it's Friday and I feel better, so I'm going to go out with my friends anyway and stay out late. Bad time to do that because, well, I know you can't do that now, but if you could, during that convalescent period, you're really susceptible to secondary infections because you're not back up to 100% yet. So your body is still fighting off whatever that first infection was, and then that makes you more susceptible to getting a secondary infection, which then also has to go through all those phases. So then it might feel like you're sick for 3 weeks instead of just a week or a week and a half. And that last period that they added on there, which I don't think I have on my list, I do, the continuation period. This would be for diseases that persist, right? So think like HIV or herpes that you have with you forever, right? Or for a continuing period of time. So you notice the blue line either goes down, period of decline and convalescence and decline, or it's going to go on, period of continuation, and that is disease specific. Common signs, common symptoms, that's just kind of a nice list. You, of course, need to remember that signs are measurable, things that you can actually measure and quantitate, or that are objective. And symptoms, notice these are all reported, self-reported, as well as more general subjective changes, right? Usually they put skin eruption, so that's like a rash, right? So they usually put rash on signs, but there it is. And then the organism has to have a portal of exit. It has to have a way to get out so that it is spread to other susceptible hosts, right? So there's just some secretions listed there, some portals of exit. This kind of ends that discussion of how an organism can cause a disease. Now, pretty much animals, including humans and arthropods, can be reservoirs. We divide up reservoirs into living and non-living reservoirs. And it's pretty straightforward, a living reservoir, an animal or a human, and we pull out humans that are actively ill, carriers, arthropods, and then we pull out for non-living reservoirs, soil, water, air, and the built environment. So we mean places where we live or we work. A reservoir has a very specific meaning. It is a place where an organism can multiply and persist. So it can divide if it's a virus, or if it's a bacteria, it can multiply and has opportunity for transmission, a way to get to its next host, right? So a living reservoir, humans that are actively ill, clearly we can spread the disease from one person to another. We're going to talk about transmission, which is different in a minute, right? We're talking about where the organism can persist. Inside people that are actively sick, inside people who might be recovered or maybe never felt sick, but are still shedding the virus. And then arthropods, which of course can transmit by biting you and then transmitting to another host. So here's some carriers, right? And the list is long. Asymptomatic, incubating, convalescent, chronic, passive carriers. And again, we all know a lot more about what an asymptomatic carrier is due to the virus pandemic that we're living through right now. But this is sort of a general term. In reality, most of the carriers are probably all of these, at least all of these first three. Most of the carriers of COVID are either asymptomatic, incubating, right? They haven't shown any sign of disease yet, but they're carrying it and they're going to get sick, or they're recovering and they're not completely convalescent yet and they're still shedding virus. We're not sure if they're shedding whole virus or if they're just shedding RNA. We don't know, but I think, of course, as time goes on, we're going to find out more and more. Different types of vectors. We refer to the arthropods that transmit as a vector. And we've got biological vectors. Those are vectors that are infected, and then mechanical vectors that are just depositing bacteria or virus on the surface of something. These are biological vectors. We're going to eat them or we're going to get bitten by them. and they're going to transmit the infection. Zoonosis is an important term to pay attention to here at this time. This refers to an infection that's indigenous to animals but can also be transmitted to humans. Most of the time we're a dead-end host and we don't contribute to the natural persistence or being a reservoir for a particular infection. Close association with humans and animals, and often affects people more that are in animal-oriented or outdoor professions that are at greatest risk. So I think about rabies, for example. That's a good example of a zoonosis or zoonotic infection. It is not an infection that is sustained in humans. It is sustained in the wild population of other animals, and then humans are an accidental host or a dead-end host, right? And you're more likely, if you work with animals, like if you work in a veterinary clinic, for example, you're much more likely to be exposed to rabies, or if you work for like animal control and you go around and remove raccoons and bats from people's houses or sheds or outdoor areas, you're going to be more likely to contract that disease. I talked about the non-living reservoirs. These are patterns of transmission for communicable diseases. The list is long, right? So this is how the disease gets from one person to another. We've got direct contact. So vertical transmission is very limited. This is just transmission from parent to offspring via through the placenta or through breast milk. Horizontal transfer is when we transfer to another person that we're not carrying as a baby. So horizontal transfer, direct contact transmission, indirect, if there's some sort of an object that the sick person touches the object and then a healthy person touches the object and gets infected. And there's some lists here, right? A fomite is an inanimate object. Vehicle, indirect, is any non-living that can infect, like air, water, soil, food. And the special category of fecal to oral, basically if you don't wash your hands after going to the bathroom, you can easily spread particular organisms that pass from the feces of one person into the oral cavity of another to cause disease. And then the vector transmission, I guess I should move those vectors to here. Yeah, that slide's in the wrong spot. All right, I'll try to fix that. That pretty much sums it up. Vehicle, fomite, oral fecal route, oil and soil can harbor infections, air, droplet versus aerosol, that's important. Here's our classic picture of the gentleman giving us a nice big sneeze. A droplet nuclei is something that usually only transmits within about 3 feet. So we actually use 1 meter as the distinction for droplet nuclei. That's why we're all staying 6 feet apart, because my droplets can only maximally travel 3 feet and your droplets can only maximally travel 3 feet the other way. So if we're staying 6 feet apart, nary our droplets will mix, right? And there's no chance I'm going to inhale droplets from you if you've had a sneeze such as this gentleman. Particles that can travel farther than a meter or 3 feet, we consider those aerosols, and the jury seems to still be out whether or not COVID can transmit as an aerosol. It doesn't seem to be unless you're in extended contact with a person, because most of the time it does appear that the mask wearing and the social distancing is preventing the transmission, even among people who live in the same house and one person is positive and other people don't become positive when they follow those social distancing rules. So that is an important distinction. Then I think I've listed Cook's postulates. This is how we determine what is the etiological agent, meaning what is the organism that causes a particular disease. We start by looking for evidence of a particular microbe in every case of the disease. We try to grow the organism. We try to isolate it from everything else. And then the classic test is to inject an animal model, either like they're injecting into the bloodstream here, or you might make an aerosol that the animal would inhale. And then we look to see the same signs and symptoms of the infection. And then we look to re-isolate the same bacteria or the same infectious agent from this second subject, this what we call a naive subject. So clearly, we can't do this test. We can't follow Cook's postulate sometimes for viruses because they're difficult to grow outside the body. And of course, we can't follow this test and inject healthy people with an agent just to see if it makes them sick. That's why we usually use an animal model. And then other times it's based on a large body of evidence. For example, with COVID, determining which virus was actually causing the signs and symptoms, the particular signs and symptoms for COVID-19 was based on basically the signs and symptoms report of huge populations, primarily from China, but then early on as well from Europe and the United States, by determining, look, these are the same symptoms we see in all these people, and we test them for this virus, they're all positive for this virus. So instead of infecting other animals to see if they get it and get similar signs and symptoms, we're going to go ahead with a fair amount of confidence and say this particular virus is the etiological agent or the cause of this particular set of signs and symptoms. Okay, so that can vary. Sometimes we do, it depends on what the disease is. You mean for a zoonosis? It depends on the disease, right? What we mean by dead-end host is we're not an active transmitter of the disease, right? It doesn't become a respiratory infection in us. We might get a skin infection instead. That could be transmitted, but very rarely to another person, right? So it's usually not the same portal of entry, and it's usually not the same set of signs and symptoms that the animal had. Okay, so we did Cook's postulates. Let's go back. Again, we're poised in a lot better space now. I can condense this whole section of the CDC and epidemiology to a much shorter lecture in some respects, because you are probably a lot more familiar with the CDC now than previously in the past other classes have been. I would point out to you that they publish a morbidity and mortality weekly report. Comes out on Friday night at midnight, or actually Thursday night at midnight, so it's available Friday for people to read. It lists all of the notifiable diseases and how many people have been reported positive for those diseases and how many people have died from those diseases by state this week compared to the whole year, compared to last year at this time. And so it's a tracking system, a methodology to try to figure out how a disease is spreading in a population. Which brings us to this, prevalence, incidence, and mortality rate. Super important words here for epidemiology. Prevalence is the total number of existing cases with respect to the entire population. So the prevalence of a disease, for example, for COVID, if you're looking at the numbers, would be both the people that have tested negative and the people that have tested positive, right? That's the best measure we have. We don't really know how many people have the virus because we're not testing every single person. Only if we tested the entire population would we know, right? The incidence is a measure of new cases over a certain time period, right? We also call that the morbidity rate. The mortality rate is obviously the number of deaths in the population due to a certain disease. And depending on what type of epidemic is being sourced here, when this is a point source epidemic, so we have one infectious event, maybe a restaurant that was serving something that had salmonella in it, and then everyone who ate at that restaurant, and then we see an increase in anyone who can relate back to that particular event. The common source epidemic, this is much more what we're seeing with COVID, right? We're seeing that many people have a infection, right? And then it's also actually a propagated epidemic because it's continuing on as these people who are infected are transmitting to new people. And of course, we would be referring to this as a pandemic all the time because it's spread across multiple continents. I would encourage you to take a look at, in your textbook, the list of notifiable diseases that the CDC reports on. I think you can clearly see, it's the very end of the chapter, I think it's on page 377. You can clearly see that they are charged with reporting on multiple diseases. This data is collected weekly from literally county health departments that report to state health departments that then report to the CDC, where it's all collated and pulled together. So it's not a perfect system, but it's a relatively good system, and it's what we have, right? And so this is how we trace epidemics and how we trace pandemics and see what is going on. That ends the material that I have